Hello everyone, I would like to welcome you to Story Time with Alice. Today we will be reading Protein Synthesis. Once upon a time, inside of a cell's nucleus, the gene encoding a protein was copied into RNA. DNA felt left out, so we decided to be embedded into the cell's chromosomes. When they soon realized that with all of the transferring going on, they made a great team. They decided to give all of their work a name. They called it transcription. The next day, someone asked, "Well, what does transcription do?" "What does it do?" they said in a shocking voice. "Transcription helps to magnify me by creating copies of RNA, which act as a template for protein synthesis," said DNA. "My copy of the gene is called mRNA," said RNA. "We are both constructed by a chain of nucleotides," said the two friends. "So I don't get it. Do you both do the same thing because you guys are best friends?" asked little Flagellum in the back. "Of course not. I differ from DNA by substituting uracil for thymine, and RNA substitutes cytosine for guanine. Also, only one strand of mRNA is needed when synthesizing proteins, and mRNA naturally exists in single-stranded forms," explained RNA. "Here, let me show you," said DNA. After transcription happens, mRNA is asked to leave the cell's nucleus. We usually let it go on the nuclear pores. I mean, it has to go all the way to the site of translation, said DNA. "Do you mean through the rough endoplasmic reticulum?" "Yes," asked Lysosome Jr. "Yes," said RNA. "Whoa," shouted the class. "He's one lucky guy," said the mitochondrion. "What happens next?" asked Flagellum. "Well, after mRNA gets to the rough endoplasmic reticulum, he is guided to the ribosomal translation machineries to meet up with his friends," said DNA. "Then our friend ribosome reads the mRNA sequence from the fifth end to the third end," explained RNA. I heard that mRNA wanted to be converted into a protein. Is that true?" said Flagellum. "Yes, it is," said DNA. tRNA was used to read the mRNA sequence, 3 nucleotides a time," said RNA. "Has anyone heard of amino acids?" asked RNA. "Well, amino acids are represented by codons, which are 3 nucleotide RNA sequences. The mRNA sequence is matched 3 nucleotides at a time to a set of 3 nucleotides in the anticodon region of the corresponding tRNA molecule," explained DNA. "So, opposite of the anticodon region of each tRNA, an amino acid is attached," said RNA. "Also, an amino acid is attached, and as the mRNA is read off, the amino acids on each tRNA join together through a magnificent peptide bond," shared RNA. Now, not everything went right during our creation of this project. There were a few problems in my nucleotide sequence, which were produced by factors called mutagens, explained DNA. "We figured out that whenever that happens, a chemical bond breaks in my friend DNA and results in loss of segments. This also happened during some replication," stated RNA. "So this process we decided to call mutation." a few types of mutation that we will briefly share with you, starting off with the first type. Point mutation is simply when a single base is replaced by a different one. This may or may not change an amino acid sequence, said DNA. Second type is an addition/deletion. This is when whole sections of DNA are added or deleted, which result in misreading a code or a loss of a set of genes. So this is point mutation. And addition and deletion. So all in all, it's very important that we work together to help take care of our project, said RNA. Does anyone have a name we could use for this? Asked DNA. How about we call it protein synthesis? Shouted Flagellum. That's perfect, shouted the boys. And everybody lived happily ever after. Thank you for joining me for Storytime with Alice. I hope to see you soon.